Scientific Officer Biology, Immunology Part-3, Notes and MCQs. These are the topics covered in this video. Antigenic determinants of immunoglobulins. Immunoglobulins, which is also known as antibodies, are glycoproteins produced by B cells that play a critical role in immune response. They can be classified based on various antigenic determinants. They are isotype, allotype, and idiotype. Isotype refers to the class of antibody determined by the constant region of the heavy chain. There are 5 main isotypes: IgA, IgD, E, G, and IgM. Each isotype has distinct roles in the immune system. For example, IgG is the most abundant in blood and provides long-term protection, while IgE is involved in allergic responses and defense against parasitic infections. Allotype are genetic variations in the constant regions of antibodies between individuals of the same species. These variations are inherited and can differ between individuals. Allotypic differences can affect the interaction of antibodies with the other immune system components, but they generally don't have a significant impact on the antibody's overall function. Idiotype, which refers to the unique antigen binding site of an antibody, which is determined by the variable regions of both heavy and light chains of immunoglobulins. This is where the antibody binds to the specific antigen. Idiotype is specific to the individual antibody and defines its antigen specificity. It's what differentiates one antibody from another in terms of the antigen it binds to. B cell receptor. The B cell receptor is a membrane-bound immunoglobulin found on the surface of B cells. It functions as a primary means for B cells to recognize and bind to specific antigens. Components of BCR includes immunoglobulin molecule and accessory molecules. Immunoglobulin molecule, the part of the receptor that binds to the antigen. And the second one is the accessory molecule, which is examples such as CD19, CD21, and CD81, which help transmit the signal into the B cell once the antigen binds to the immunoglobulin. And these two parts of BCR, B cell receptor. Then the topic is monoclonal antibodies. Monoclonal antibodies are identical antibodies produced by a single clone of B cells, all of which recognize the same specific epitope on an antigen. They are highly specific and uniform. The technology that used to produce monoclonal antibodies is called the hybridoma technology, which is invented by Kohler and Milstein in the 1970s. Hybridoma technology is briefly explained here, and different steps are there. The first step is immunization. Immunization means the mice or other animals are immunized with an antigen to stimulate an immune response. Then the second step is cell fusion. B cells from the immunized animals are fused with the myeloma cells or the cancerous cells to create hybridoma cells. These hybridomas have the ability to produce antibodies and proliferate indefinitely. And next is selection and screening. And the step is cloning and expansion. Selected hybridomas are cloned and expanded to produce large quantities of monoclonal antibodies. Then lastly, antibody purification are done. And these monoclonal antibodies represent a powerful tool in modern medicine, offering targeted therapies with the potential for high efficacy and specificity. Clinical uses of monoclonal antibody. First one is diagnosis. It helps to detect pathogens, used in various diagnostic assays to detect the presence of specific pathogens or biomarkers. And the second use is the immunomodulation, immune system regulation. Some monoclonal antibodies are used to modulate immune responses in conditions like multiple sclerosis and inflammatory bowel diseases. Another clinical use of these are therapeutics. Used in therapeutics, for example, cancer treatment. Monoclonal antibodies can target cancer cells specifically, marking them for destruction by the immune system or delivering cytotoxic agents directly to them. Examples include the trastuzumab for HER2 positive breast cancer and rituximab for certain lymphomas. Autoimmune diseases. Monoclonal antibodies can block specific immune pathways involved in autoimmune diseases. For example, the adalimumab targets TNF alpha in rheumatoid arthritis, and it can be also used in infectious diseases like COVID-19. MCQs related to the topics. Which of the following immunoglobulin isotypes is most abundant in blood and provides long-term protection? IgG. What term describes genetic variations in the constant regions of antibodies between individuals of the same species? Allotype. The unique antigen binding site of an antibody determined by the variable regions of the heavy and light chains is referred to as idiotype. Which component of the B cell receptor is responsible for recognizing and binding to specific antigens? Variable region. Which accessory molecules are involved in signaling following antigen binding by the B cell receptor? CD19, CD21 and CD81. The B cell receptor is primarily composed of immunoglobulin molecules and signaling proteins. In hybridoma technology, what type of cells are fused with the B cell to produce monoclonal antibodies? Myeloma cells. Which process is used to select hybridomas producing the desired antibody after cell fusion? Screening. Which monoclonal antibody is used for targeting HER2 positive breast cancer? Trastuzumab. Monoclonal antibodies can be used in the treatment of autoimmune diseases. by blocking specific immune pathways. Which of the following monoclonal antibodies is used to neutralize the SARS-CoV-2 virus in COVID-19 treatment? Casirivimab and imdevimab. Which of the following is not a typical clinical use of monoclonal antibodies? Allergic reactions. In diagnosing infectious diseases, monoclonal antibodies can be used to detect specific pathogens or biomarkers.